The fluid mosaic model of membrane structure. Proteins in the cell membrane. Membranes have many associated proteins that can be classified according to where they are located with respect to the phospholipid bilayer. The proteins we'll talk about in the following slides are peripheral proteins, transmembrane proteins, and integral proteins. Peripheral proteins are anchored on one side of the membrane, but are not embedded in the membrane. Some peripheral proteins have a tail of hydrophobic amino acids that protrudes into the fatty acid layer anchoring them, which is what you see here. Other peripheral proteins are bound to a domain of an integral membrane protein that protrudes from the membrane, which is what you see here. So these are anchored to these other proteins. Integral proteins are embedded in the membrane. So right here, this is embedded in the membrane. So are these two. Embedded in the membrane. They have hydrophobic amino acids facing the interior of the membrane, anchoring them in the hydrophobic space. Transmembrane proteins span the entire membrane, like this here and here, and have hydrophilic domains facing both the extracellular and intracellular environments. They often form selective channels, which you can see here, across the membrane. Glycoproteins and glycolipids. Glycoproteins and glycolipids have attached chains of sugars that extend out of the cell. This prefix glyco means sugar. These sugars are used for communication with other cells. So you can see this entire thing here is a glycoprotein. So it's a protein in the membrane that has a sugar chain attached to it. And then glycolipids are these, you can see these little lipid tails here that are inside of the membrane. Just as an insect might use antennae to gather information about its environment, cells respond to the environment after their glycolipids or glycoproteins have specific interactions with other molecules. So these sugar chains on the outside here of either the glycolipid or the glycoprotein have the ability to then interact with the environment outside, and those specific interactions are communications. Membrane protein interactions with ECM and cytoskeleton. Cells are in constant communication with and responding to their internal and external environments. Certain transmembrane proteins, integrins for example, interact with proteins of the extracellular matrix, or ECM, as well as the internal cytoskeleton. It is one path of communication between a cell and its environment. So let's take a look at this image of this membrane we have here. So we have our integrin, our transmembrane protein. On the inside of the cell, we have these microfilaments of the cytoskeleton, okay? And then on the outside, coming from this integrin protein, we have a sugar, a polysaccharide, and off of that, some protein, some carbohydrates, okay? And that's all wrapped up and interacting with the collagen fibers and everything in the extracellular matrix, okay? So all of those things are constantly working together in communication with each other, responding to all of those different environments as a way to communicate between the inside of the cell and the surrounding environment. The ECM, the extracellular matrix, is made of cell-derived proteins and provides tissue structure. Just as a brick wall is more sturdy with bricks embedded in cement, a tissue is more sturdy if its cells are embedded in their extracellular matrix. Fluid mosaic model. While the ECM and cytoskeleton help to give some cells stability, phospholipids, cholesterol, and some proteins of the plasma membrane flow freely. Therefore, the plasma membrane is described as a fluid mosaic, like mosaic tiles that are moving in grout. So here's what we have. We have our extracellular fluid. This is the outside of the cell. The cytoplasm inside of the cell. We have some transmembrane channel proteins, some peripheral membrane proteins, an integral membrane protein, right? We've got a glycoprotein, a glycolipid on the outside, helping us do that communication. And then we can see some of these molecules moving through. Here's a cholesterol molecule and some other molecules that can freely move throughout the membrane. So we have this fluid mosaic structure. Many proteins can also move within the membrane while forming connections with structures inside and outside of the cell. One landmark experiment in this fluid mosaic model came from scientists Fry and Edidin. They carried out a landmark cell fusion experiment in 1970, which demonstrated the fluidity of the cell membrane. Their work with human and mouse cells supported the fluid mosaic model of membrane structure. So let's look at this experiment a little more. So what Fry and Edidin did was they labeled mouse cell membrane proteins with a red tag. So you can see like those outer cell membrane proteins, they labeled them with a red tag. And human cell membrane proteins with a blue tag. So the human cells were blue, the mouse cells were red. The cells were combined in the presence of a virus, and the purpose of that virus, the virus causes cell membranes to fuse, okay? And so that fusion, through the help of that virus, created some, so that some of the fused cells were these human-mouse hybrids because of the fusion of the membranes. And under the microscope, they observed that the labeled proteins intermixed and were evenly distributed over the hybrid cell after 40 minutes. So when we get these fused mouse-human hybrids, we can confirm their fluidity because after a period of time, they are evenly distributed over that hybrid cell. The proteins moved in the plane of the membrane so that they were evenly mixed. Cholesterol in the cell membrane. Cholesterol is a lipid found in cell membranes. The presence of cholesterol stabilizes cell membranes by adapting their flexibility as a function of temperature. Cell membranes must maintain fluidity and flexibility to carry out their functions. Cholesterol decreases flexibility at high temperatures, preventing excess flexibility. At low temperatures, cholesterol adds more flexibility to membranes. In the image, you can see this molecule right here is the cholesterol in the cell membrane. Fatty acids in the cell membrane. In addition to the cholesterol, another factor that influences fluidity of the cell membrane is the presence of fatty acids. Saturated fatty acids are solids at room temperature, while unsaturated fatty acids are liquids at room temperature. The ratio of unsaturated to saturated fatty acids impacts the fluidity of membranes because saturated fatty acids pack closely, while unsaturated fatty acids are spaced apart, which would lead to an increase in fluidity. All right, here's example 1 for the fluid mosaic model. Fry and Eddington carried out their first experiment at 37 degrees Celsius. A, predict what happened when they carried out the experiment at 20 degrees Celsius and explain your reason. And B, predict what happened when they carried out the experiment at 4 degrees Celsius and explain your reasoning. So, what would we expect to happen at 40 minutes if we lower the temperature a little bit and then lower it a lot more? All right, so at 37 degrees, Fry and Eddington saw even mixing of those mouse-human hybrids, right? What would happen at 20 degrees Celsius? So it's a decrease in temperature. And then explain your reasoning. So because membranes are less fluid at lower temperatures, after 40 minutes, the membrane proteins would be mixed, but not completely intermixed, as we saw at 37 degrees Celsius. Now, what about at 4 degrees Celsius? Remember that the freezing point of water is 0 degrees Celsius. So this is 4 degrees Celsius, very, very low temperature. What would happen? At 4 degrees Celsius, mouse and human membranes would be solid. What happens if they're solid? There's no fluidity at all. All biochemistry would be inhibited and the cells would not be able to fuse. Example 2, fluidity of cell membrane. Fish are ectothermic animals, meaning that their body temperatures are influenced by their environments. Mammals like us are homeothermic, which means we maintain stable body temperatures. Predict whether Arctic fish, such as the Arctic char shown in the image, have high or low concentrations of cholesterol and saturated fatty acids in their cell membranes when compared to mammals. Explain your reasoning. Okay, so these Arctic char obviously live at very cold temperatures, very low temperature, okay? And those temperatures are influenced, the temperature inside the fish is influenced by the low temperatures that they live in. And so we need to figure out what would cholesterol do in a cell membrane and what would saturated fatty acids do in a cell membrane at low temperature. So remember that cholesterol increases fluidity at low temperature. And saturated fatty acids are solids at room temperature, so even lower temperatures than room temperature would still be solids, okay? So at a low temperature where these fish are living, they need their membranes to still be fluid. So we need to make sure we're producing or they have these membranes to maximize their membrane fluidity even at the low temperatures. So Arctic char would have higher concentrations of cholesterol, right? Because an increase in cholesterol would increase the fluidity at low temperatures, but lower concentrations of saturated fatty acids, right? Because that would make them less fluid. At low temperatures, both the presence of cholesterol and unsaturated fatty acids will increase the fluidity of cell membranes.